Noor Zayanah is currently a research assistant at the Viral Research and Experimental Medicine Center in Duke-NUS Medical School, Singapore. She recently completed her master's by research at the University of Bristol under the supervision of Associate Professor Dr. Laura Rivino, where she studied the responding T cells in acute dengue infection. Her research interests include T cell responses in flavivirus infections and early phase vaccine clinical trials. And her title, as you can see here, is The Role of Skin-Based T Cell Immunity in Acute Dengue Infection.
Right, thank you so much for the introduction, and thank you, ProImmune, for inviting me to speak. I'll be sharing work that I did for my master's project, and so it's about the role of skin-based immunity in acute dengue infection. And perhaps I'll just take you through first on the disease burden worldwide and in Singapore. Dengue is widely transmitted across tropical and subtropical regions, placing about 2.5 billion people at risk of infection. And over the decades, the number of dengue cases has increased over wider geographic regions. From the years 2000 to 2008, we saw that there were more than 1 million cases recorded across 69 countries. And in 2023, there were over 3.7 million cases already been reported from 70 countries globally. In Singapore, where my study is based at, dengue is an endemic disease. So, several epidemics have occurred frequently, such as in 2020, shown in yellow, where it hit record-breaking cases since dengue was first reported in Singapore. In 2020, more than 35,000 cases were recorded that year. And we saw another epidemic again in 2022, shown in blue. So dengue virus is a positive sense single-stranded RNA virus from the flavivirus genus, which includes other viruses such as Zika, yellow fever, West Nile, and it has 4 distinct serotypes known as dengue 1 to 4. Hence, a person could potentially be infected with dengue about 4 different times. It is transmitted via the Aedes mosquito. And I thought I'd show you some posters that you might find around Singapore, because here we are pretty big on trying to get everybody to play their part in eradicating the mosquito population, since they can thrive in urban environments as their breeding spots. Currently, there are no licensed therapeutics to treat dengue, and the licensed vaccines in the market do not elicit equal protection against all serotypes and have safety issues. So how does the disease progress in humans is that following the bite of the Aedes mosquito, there's an incubation time of about 2 to 3 days before there's detection of viremia and symptoms. And symptoms may include rash, muscle aches and pain, nausea and vomiting. And high viral load and longer duration of viremia is strongly correlated with increased disease severity. But when viremia falls, that's also when severe dengue manifestations such as plasma leakage, shock, and organ impairment can occur. It is happening at the time when T cells start to increase and coinciding with inflammatory cytokine production. And so my study is looking at this time frame here when T cell responses are high. So why are we looking at T cells in dengue? Because their role in dengue is not well understood yet. And there's generally about 2 different opinions on it. The first being that T cells play a pathogenic role, mostly described by the original antigenic sin hypothesis. So in a similar way that you might think about antibody-dependent enhancement and antibodies, the original antigenic sin hypothesizes that memory T cells from a previous infection are unable to efficiently clear the dengue-infected cells of a secondary heterotypic infection, because the memory T cells have poor affinity to the secondary virus due to the different T cell epitopes. But more studies have pointed to the protective role of studies. So for instance, more studies have pointed to the protective role of T cells. So for instance, a study showed that Children with subclinical dengue had higher frequencies of dengue-specific CD4 and CD8s compared to symptomatic children. And people with HLA types that are less susceptible to severe dengue have higher magnitude and more polyfunctional T-cell responses, suggesting that a vigorous T-cell response leads to better protection. And in some mouse models, T-cells have been described to be necessary for viral clearance through the production of pro-inflammatory cytokines and cytotoxic activity. So we're looking at the skin because essentially that's the site of infection. That's the first point of contact where the infected Aedes mosquito takes a blood meal from the humans. The dengue virus is released, which then targets antigen-presenting cells such as Langerhans cells in the epidermis or other dermal APCs. And these APCs will travel to the draining lymph nodes where the T cells are primed. And these T cells will then express a marker called CLA, or cutaneous leukocyte-associated antigen, which facilitates homing of the T cells back to the skin. The T cells may also circulate to the blood where they may encounter the virus there, but a lot of studies in dengue have looked at the blood, whereas the T cells in the skin are less characterized. So in the skin here, they have the potential to become tissue-resident memory T cells, where they may permanently reside in the skin and patrol for antigens and may directly target virus-infected cells. So therefore, the aims of my project was to characterize the T cells in the skin and blood during acute dengue infection and to understand the T cell correlations with clinical outcomes. We recruited over 30 dengue patients and 10 healthy donors for this study and collected information regarding a couple of clinical parameters related to dengue for us to make associations with disease severity. The dengue cases in Singapore are rather mild, so we don't get a lot of severe dengue cases. So we were looking at other parameters on top of the WHO classification to gain understanding on the disease outcomes. And I just want to highlight here these two parameters, which I will mention again in later slides. We have about 40% primary infected patients, meaning patients who caught dengue for the first time, and 60% secondary infected patients, meaning those who caught dengue for the second time, most likely from a different serotype of dengue now. And usually patients who have a secondary infection report more serious or more severe dengue manifestations. Whereas for the hospitalization status, about 20% are outpatients and the remainder 80% are inpatients. So how the study was carried out was that the dengue patients and IgG negative healthy volunteers come for 2 visits. At the first time point here, we collected blood for preliminary work, such as checking their serostatus, their IgG levels, and as well as their HLA type. But the bulk of the analysis comes from the second visit, which is days 7 to 10 from fever onset. And here we collected matched PBMCs as well as skin blister fluid from the same donors. So to collect this skin blister fluid, basically a blister is induced on the forearm of the donors through a negative pressure pump, and the donors wait for about 2 to 4 hours until this bump here is formed, which is when the epidermis and dermis layers have separated. And that area gets filled with fluid and cells. And then, they go back home or back to the wards with like this bottle cap on their forearm for about 18 to 24 hours. And the next day, the fluid is aspirated using a needle and into an Eppendorf tube where I spin down to get the cell pellet. And in my study, I'm interested in the immunophenotypes and the potential function of the T cells from the matched PBMCs and cells from the skin blisters. And I stained them with a 16 to 17 color flow cytometry panel, and I'm looking at various markers that are related to these different characteristics of T cells. and analyze them using UMAP as well as phenograph, which is an unsupervised clustering algorithm. So my first aim was to characterize the T cells in the skin and blood. And here we are looking at a global view of total CD3 cells from dengue patients and healthy volunteers, separated into different clusters based on their different immunophenotypes. So the CD8 cells take up the islands on the left here and the CD4 on the right. And we can see that there are distinct populations of blood T cells and skin T cells. So the blood T cells sort of clustered towards the bottom of the graph, and the skin clusters at the top, implying that they have different characteristics. I then distinguished further between the dengue patients and the healthy volunteers, and we can see that there are different distributions of these T cell clusters in these four different compartments. Some clusters, may overlap with others, but some are more unique to certain compartments, implying that there are different immunophenotypes of these T cells in the different compartments. To take you through further, the frequencies of the clusters and their relative MFI of the various cell markers are depicted in this heat map. So to take you through, clusters that are in yellow or bright green has higher frequencies than the dark blue ones within that column. And the top half of this heat map shows clusters that are more enriched in the skin compared to the blood. But what is particularly interesting to me were the clusters that are even more so enriched in the skin of dengue patients compared to the healthy donors, such as clusters 2 and 9 here, which tells us that the T cells in the skin of dengue patients have relatively high expressions of proliferation marker Ki67, activation marker CD38, PD1, tissue resident marker CD69, as well as coenzyme B, indicated by the red boxes compared to the blue boxes, which implies lower expression. And to further confirm the characteristics of dengue-specific cells, we stain the PBMCs and skin cells with the proimmune Pro5 MHC class 1 pentamers. And here's a representative figure from one patient here who was stained with 2 pentamers that were specific for their HLA type and the current serotype of dengue. So to take you through, this is the global view of the T cells from this one patient here. And again, we see quite distinct populations of the blood T cells and the skin T cells. So the blood takes up more of the top half of the graph and the skin towards the bottom. And when I overlaid the dengue-specific pentamer cells in green here, it corresponds to the cluster 3 in orange here, which we can see is more numerous in the skin than in the blood. And so looking at the characteristics of these dengue-specific pentamer positive cells, we see that they have relatively high expressions of activation markers, as well as CD69 compared to other clusters. And again, we see there's higher frequencies of these dengue-specific cells in the skin than the blood of this patient. We did similar stainings for 8 other patients. And generally, we do see higher percentages of dengue-specific cells in the skin than the blood, though this did not reach statistical significance. And then at this stage, it is I face the challenge of getting more patients with a positive staining for this dengue-specific cells. So I was wondering, how else can I look at responding T cells in acute dengue infection? So based on Laura Rivino's previous work, she saw that dengue-specific cells from PBMCs of acute dengue patients had high Ki67 as well as CLA expression, but these markers were not highly expressed in non-dengue-specific cells and bystander-activated T cells. So we adopted this Ki67 CLA double population as a proxy to study dengue-specific T cells in the blood and in the skin. And indeed, we do see that this population is higher in the skin than in the blood, but also significantly higher in dengue patients than healthy volunteers. So then I further drilled down on this Ki67CLA population to look at the different markers of interest and their characteristics. Firstly, tissue resident marker CD69 is higher in the skin than the blood in both CD4s and CD8s, which is quite expected. Granzyme B was higher in the skin than the blood of CD4 cells, but the expressions was similar and quite high between both blood and skin and CD8s. And similarly, skin CD4 cells were more activated than the blood, but quite comparable in the CD8s. With this in mind, we then asked whether T-cell responses in the skin and blood correlated with clinical outcomes. And what struck out to us was hospitalization and serostatus. We saw that increased Ki67CLA cells in the skin, it correlated with outpatients as well as primary patients. So in other words, having high frequencies of this population in the skin correlated with milder disease, but this was not seen in the CD4s in the blood. And for the CD8s, we see similar high frequencies of Ki67CLA in the outpatients for both the blood and the skin, but no significant differences were observed between primary and secondary infection, though maybe I might argue that perhaps there's a bit of that trend there in the skin. So then, it appears that high frequencies of Ki67CLA in the skin might be associated with milder disease outcomes. So to summarize my work, we saw that T cells are distinct in the skin and blood, shown by the separation of clusters in the UMAP because they have different immunophenotypes. There's more dengue-specific T cells in the skin than the blood, and these dengue-specific cells are activated, they're proliferating, they express CD69 as well as granzyme B, and higher percentages of this Ki67CLA population in the skin is associated with milder clinical outcomes. All right, thank you for listening to my talk. I'd like to say thanks to my lab in Bristol, as well as Duke-NUS, as well as the clinical team at NCID Singapore. Thank you for listening.
Great, thanks, Noor. Wonderful talk. Are there any questions from the audience? Oh, yes, Professor Choi, go ahead.
Hi, today I just wonder what are the statistics of the HLA, the types of the patients. So are there people who are more susceptible to the dengue virus infection or something like that?
I can't say the HLA types at the top of my head right now, though I can say that based on previous work by Daniela Westcott group, she said that HLA-A types usually have like higher frequencies but low polyfunctional responses. So they are the ones who are high frequencies, low polyfunctional, so they are more susceptible to dengue, whereas certain types of HLA-Bs was the opposite trend. But I don't know for sure which HLA types are more susceptible to others.
Okay, thank you.
Other questions?
Hi, I have a question. Thank you for this great talk. I wanted to ask, you mentioned that increase in skin resident CD4 positive T cells, your cells of interest, increase in their numbers correlate to better recovery in the patients?
It's an association where we see like outpatients had higher frequencies of these Ki67CLA in the CD4s.
Okay, so do you also see association with symptoms? For example, skin manifestation of dengue, for example, rashes and stuff. Do you see correlation with T cells and the skin?
Yeah, so we did look at rash, but we didn't see any differences in with like patients with or without rash.
Okay, I have a last follow-up question. Thank you for answering the previous questions. Is it specific to a serotype of the dengue virus? Do different types would lead to different induction degree of these T cells?
I don't know. The cohort that I have are mostly dengue 2s and dengue 3s. But okay, so when I do look at dengue 2s versus dengue 3s, they don't have differences. But I also might think that, you know, whether they are primary or secondary infection might also be a confounding factor. So I think that's a little bit difficult to tease apart. Yeah.
Thank you so much. It's a very interesting study. Thank you.
Great. Thanks, Noor, for that wonderful talk. We'll move on to our next speaker now.